To measure spatial receptive fields for the cortical activity map, we use a locally sparse noise stimulus. Usually, a sparse noise stimulus consists of a small white or black spot that are briefly flashed on a mean luminance gray background. This has been very effective at mapping the on and off subunits of cells' receptive fields. However, to sample all of the possible locations sufficiently, which in our case is roughly 450 locations, can take a very long time. Because of this, we developed a modified version of this stimulus that we call the locally sparse noise. For this stimulus, we present multiple 4-degree spots, both white and black, at the same time, but we make sure that no two spots are located within 20 degrees of each other. We can consider all of the stimulus frames when there is a white spot at one location, such as this one. While each frame has an array of different spots, they are arranged differently each time. When we average these together over 100 frames in total, we see first that there's a very clean exclusion zone of 20 degrees around this location where no other spots have been located. Beyond this, we see that the other spots have averaged together to create noise. Because of this, we believe that if, for a given frame, the presence of other spots does affect the response of the cell, that when considered in total, this effect will average away. Now we can look at the response of a cell to spots that are flashed in a given location. We've plotted here is the mean change in fluorescence for a given cell when there is a white spot presented, or the on stimulus is plotted in red, or when there is a black spot, or the off stimulus plotted in blue. We see that for this location there's a very strong on response, but no response for the off stimulus. We can map this for each location in space and create a map of the cell's response, where we see that there's a small region in space with a very strong on response, which we call the on subunit. Alternately, we can represent this as a heat map, where for each location in space, the intensity on the map indicates the size of the response of that stimulus, for both the on and the off. In some cases, the mean responses can be fairly noisy, so we have done statistical tests to determine the pixels that have a significant response. Our visualization shows only the pixels that have a significant response, again showing the on responses on the left and the off responses on the right. This is a cell that has an on subunit but no off subunit. The receptive fields of our image cells can vary quite a bit. Here we see many examples of receptive fields recorded in a single experiment, showing a variety of receptive field structures. You will notice that there are some cells that don't respond to this locally sparse noise stimulus, that is, cells that don't have a receptive field. When we overlay the receptive fields of all of the cells from this experiment, we see that they overlap. Here, we represent the population receptive field, which is the sum of all the receptive fields measured in this one experiment. This plot can be used to infer the region of visual space that is relevant for all of the cells in this experiment.